Hi, welcome to another episode of Medicine Simplified. In this video, we will know about Ninhydrin test. Coming to the introduction, Ninhydrin test is a chemical test used to identify amino acid, peptides, and proteins. It works by reacting these substances with ninhydrin to create purple color called as Rohmann's purple. This test is used in various fields like biochemical analysis, forensic science, pharmaceutical testing, and food chemistry. Coming to the principle of test, the test involves a series of chemical reactions where ninhydrin reacts with alpha amino group of amino acids, and this process is called as oxidative deaminization, which releases ammonia, carbon dioxide, and aldehyde and hydrin dantin. The ammonia then reacts with another ninhydrin molecule and hydrin dantin to form Rohmann purple. In simple words, when ninhydrin is added to alpha amino group, there will be release of ammonia, carbon dioxide, aldehyde, and hydrin dantin. The ammonia present in sample reacts with another 9-hydrin molecule and hydrodantin molecule to form Rohmann purple, which indicates presence of amino acid. Coming to the procedure of 9-hydrin test, first step is preparation of reagents, which involves preparing a 2% 9-hydrin solution in a solvent like ethanol or acetone, a 1% solution of amino acid in distilled water prepared if testing for specific amino acid. If we are using it for identifying proteins, then we will make 1% solution of protein in distilled water. The second step is reaction setup, adding few drops of 9-hydrin solution to sample in test tube. Third step is heating the test tube in water bath for about 5 minutes and observing any color change. As you can see in this picture, change of solution into purple color indicates amino acid presence, which is positive 9-hydrin, and no change in color indicates absence of amino acids, and test is negative. Coming to the results, a positive result is indicated by deep blue or purplish color, signifying the presence of amino acids, peptide, proteins, or other compounds with primary or secondary amino groups. The intensity of purple color corresponds to concentration of these substances. More concentration leads to more intensified purple color. Some amino acids like proline, hydroxyproline, and asparagine can produce different colors. No color change indicates negative results. Coming to the application of 9-hydrin test, in biochemical analysis, it helps in detecting and quantifying amino acids and proteins. In forensic science, it helps in detecting latent fingerprints. In food chemistry, it helps in estimating protein content in foods. In paper chromatography, it helps in visualizing amino acids and peptides. If you like this video, please like and share, and don't forget to subscribe our channel. Thank you.